All right, today I'll be talking about the science behind RNA extraction of organs. I'm not going to go in depth into what RNA is and the whole big picture because that's a whole lesson in itself. This video is strictly for just understanding the science of RNA extraction, what it is, why we do it, what does each step mean. All right, first step, cell lysis. So in the protocol, it'll say first, put the tissue sample in a microcentrifuge tube using around 1.5 ml. This is dependent on what organ you have. So, for example, if you have brain tissue, this will work out because brain tissue is pretty easy to break up when you put it in a homogenizer with trizol. But if you have like a thick tissue like a pancreas, you're gonna probably need a bigger tube and transfer the components into another microcentrifuge tube because the 1.5 ml is gonna be way too small. You're gonna messy. Just get if you have a big piece of tissue, just get a bigger tube, lyse it, and then transfer it to another tube. All right. So first, put tissue sample into a microcentrifuge tube, 1.5 ml. Add 1,000 microliter of trisol, and use the homogenizer on the sample until you no longer see the tissue. Um, the homogenizer is basically this apparatus right here, and in the next slide we're going to go in depth to why is these two steps important. So why is this important? The point of homogenizer over here is to mechanically break up the tissue, right? It's the homogenizer basically has these little plastic pointy thingies in the end, and it spins really fast. It basically spins really fast, and just cuts open the tissue and breaks it apart. Second, we use trizol, which is a mixture of multiple chemicals, to which are there are noteworthy are phenol and guanidine thio. I always have problem with this name. Guanidine isothiocyanate. Guanidine isothiocyanate has two main functions. One, it helps cut the cell membrane of cells. Two, it helps denature RNAs. RNase is an enzyme, and the point of RNase enzyme is basically, as the name sounds, is to degrade RNase. The problem with RNase is that it's pretty common. It's rarely found everywhere in the air, so we have to destroy the enzyme so it doesn't affect our RNA concentration. The whole point of RNA extraction is um, is to extract RNA. So I made a mistake on this slide. I was just trying to give credit to the company which I got this picture from. So please don't worry about this. All right. So what is the biochemically? What does guanidine isothiocyanate do? It disrupts the hydrogen bonds in proteins. And remember, if we disrupt the hydrogen bonds in proteins, this will disrupt the native state and denature them. Um, so remember, there's proteins on the cell membrane, right? So what guanidine isothiocyanate does, it will disrupt certain protein channels, integral proteins, and if these proteins in nature, this will cause an inflow of fluid into the cell. Inflow of fluid will cause the cell to expand and eventually break open. And when it breaks open, we're releasing all of its organelles. All these intercellular components are released. We have DNA being released. We have RNA released. We have proteins, etc. Um, for legal purposes, I got this picture from this study. This is not my picture. If you want to read more about the study, please click on the link over here. It's not my picture. All right. Next step is creating acidic medium and centrifugation. We're going to add 200 microliters of chloroform. We're going to vortex our microcentrifuge tube with the trisol and the chloroform until we see a strawberry smoothie color. Then we're going to centrifuge the tube for 12,000 revolutions per minute for 10 minutes at 4 degrees Celsius. What's the purpose of this? So we add a 5-to-1 ratio of trisol and chloroform. As you can see, we added 1,000 microliters of trisol and 200 microliters of chloroform. This creates an acidic environment. Why is this important? All right, so when we centrifuge, we get two layers of solution, the aqueous and the bottom layer. And here it's called the, well, this they made a mistake. It's supposed to be organic phase, but we have the bottom layer and the top layer, and then we have this interface. All right. The aqueous layer is usually clear, and that's where the RNA is going to be. The bottom layer is usually where some DNA, some protein, and lipids, basically the hydrophobic nonpolar stuff is going to be. So the reason why we do this 5-to-1 ratio is we want to create an acidic environment. We're creating an acidic environment because we're trying to increase the number of hydronium ions in solution. If we don't increase the number of hydronium ions in solution, the aqueous phase is going to contain both DNA and RNA. Remember, we don't want DNA. We're only focused on RNA. We're trying to detect RNA only. So we want to separate RNA and DNA. So what these hydronium ions do is they bind to the negatively charged phosphate groups in DNA, and this neutralizes the negative charge, makes them less polar, and makes them drop down to the interface. That's how we somewhat separate them. The RNA is more polar than DNA, just because, as you can see, we have these two hydroxy groups, while DNA only has this one over here. That's why it's more polar. Yeah, so basically, the bottom layer should contain your more hydrophobic substances like protein and lipids. The interface should contain the DNA, and RNA should be your aqueous layer, your upper layer. And the reason why we create acidic medium is because we're trying to make DNA and RNA separate, and the only way to do that is to make DNA less polar. Next step is to collect the aqueous layer we just separated from the centrifuge and transfer to another micro centrifuge tube. We're going to discard the previous tube into the waste can in the hood. We discard it because it contains trisol, and if we're using trisol, it's always going to be the hood, so you can't put a hood chemical in regular garbage. You have to put it in the hood can, or whatever you call it. We're going to add 500 microliters of isopropanol, vortex it, then we're going to leave the sample at room temperature for 30 minutes, and then after 30 minutes pass, we're going to centrifuge again for 12 revolutions per minute for 10 minutes at 4 degrees Celsius. I apologize, I got a tongue twister. All right. Why is this important? Isopropanol in conjunction with centrifugation will cause the RNA pellet to form in the bottom of the tube. Isopropanol reduces the solubility of RNA, causing the formation of pellet, but the pellet also contains impurities as well. Next step is the ethanol wash. We're going to discard the isopropyl supernatant, and we're going to add 1,000 microliters of 75% ethanol, then centrifuge at 12,000 revolutions per minute for 5 minutes at 4 degrees Celsius. Why is this important? The impurities will be extracted from the pellet and dissolved into the ethanol, thus cleaning our RNA sample more. Next step, we're going to discard the ethanol supernatant, add, depending on the tissue, like for brain tissue, 50 microliters of RNA is fine. For pancreas or liver, you probably 100 microliters. We're going to discard the ethanol supernatant, add 50 microliters of RNA-free water, and store it in a -7 degrees fridge